Hi all, in this video we are going to talk about MHC Class 2 or Major Histocompatibility Complex Class 2 processing or mechanism. MHC Class 2 is expressed only by antigen presenting cells such as dendritic cell, B cell, macrophages and so on. So now let's look at the processing. This is an antigen presenting cell. For example, let's take it as a macrophage. Now outside this macrophage, that is in the extracellular fluid, there can be pathogens like bacteria. The macrophages, knowing this as an antigen, will engulf this bacteria through a process known as phagocytosis. In this cell, we have an endoplasmic reticulum, to be specific the rough endoplasmic reticulum with the ribosome where the protein packing occurs. Then we have the Golgi apparatus. Most importantly, this cell have lysosome. This lysosome has an acidic environment. Now this lysosome fuses with the engulfed bacteria. The fused part is called the phagolysosome. Due to the acidic behavior of lysosome, the bacteria is broken down into peptides. The ribosome in the rough endoplasmic reticulum are synthesizing MHC class 2 molecules. MHC class 2 is made up of alpha 1 and 2 domain and beta 1 and 2 domain. The MHC class 2 have another protein called II protein or the Cd4 protein, which will prevent other endoplasmic proteins from binding to MHC class 2 groove or binding site. This II protein also acts as a chaperone, which help in bringing the MHC class 2 protein out of the endoplasmic reticulum in an endosome through the Golgi apparatus. The endosome formed is slightly acidic, which will degrade the II protein, leaving a clip at the binding site of MHC class 2. It do not allow any antigen or peptides to bind to the MHC class 2 molecule. Another protein called HLA-DM interact with the MHC class 2 and releases the clip from the binding groove. Now the phagolysosomes containing the bacterial peptides then can fuses with the MHC class 2 endosome. This will allow the bacterial peptides to bind to the groove of MHC class 2 molecule. After a specific antigen binded with the MHC class 2, the macrophages or the antigen presenting cell will express it on its cell surface. Now the expressed MHC class 2 molecule will wait for the naive Cd4 T-cell. The naive Cd4 T-cell is MHC class 2 restricted. That means if Cd4 T-cell have to be activated, it should interact with an MHC class 2 molecule. Now this is our naive Cd4 T-cell. This Cd4 T-cell has a receptor known as Cd4 receptor, which checks whether the expressed molecule is an MHC class 2 or not. The Cd4 receptor segments are made up of D1, D2, D3 and D4 domains. It will bind to the alpha portion of MHC class 2. If it's completed, the Cd4 T-cell have another receptor called TCR, the T-cell receptor. The TCR checks the antigen, that is, the groove or the binding site of TCR is specific for each antigen. This will trigger a cascade of events inside the naive Cd4 T-cell called the signal transduction. This will lead to the activation of naive Cd4 T-cell to become a T helper cell. Now the T helper cell can release cytokines, interleukins and other things which will help to induce other cells which help to combat the pathogen outside the cell, and thus the pathogen get destroyed. If you like this video, don't forget to like this video and subscribe our channel. So thank you, have a wonderful day, see you in the next video.